Hi class, welcome to the Mod 7 lecture. The topic is cells, and we're going to learn about the different structures that are present in cells. Before we begin, I'm going to just roll away here in my office chair. Ta-da! There's Meeple. He likes to hang out by me when I'm working. All right, so here's our slides for Mod 7. Again, this content comes from Chapter 4 of your textbook. This is one of the longer slide sets that we'll go through all semester. I apologize, there are many different structures that we need to learn about in order to understand the structure of a cell so that we can move forward in the next coming chapters in learning about the different functions of cells as well. So we will spend a little bit of time talking about the basic features of cells. We'll review the differences and similarities between prokaryotic and eukaryotic cells. From that point forward, though, once we move into the second section of this chapter in your textbook or of this module, we'll be focusing on eukaryotes, since eukaryotes are the only type of cell that has organelles, including the nucleus. So we'll learn about the nucleus and ribosomes, the endomembrane system, the organelles that are necessary for processing energy, and then also the cytoskeleton and different structures that are present on the surfaces of cells that allow them to communicate with one another. All right, so first of all, let's look at the relative size of a cell. So most plant and animal cells, right, so that would include us, we're animals, are between 10 to 100 micrometers in size. That's really small. We're not able to see it with the naked eye or even with reading glasses like I have on. We need to go to a more powerful tool, the compound light microscope, in order to visualize a plant or animal cell, and also bacteria are visualized that way. So another way to think about that is that human cells are close in size to most bacteria. They're a little bit bigger than most bacteria. However, if we move to the smaller size of this graph, where the viruses are, we've got some of our molecules, so proteins, lipids, these are even smaller. We're not able to see these structures with even the light microscope. Instead, a more powerful microscope that is able to see more is needed. And this is the electron microscope. And it's even possible to see the atoms and different components of cells with another type of microscope. So it's important to select the right tool for the right job. It would be incorrect for us to make any type of statement about visualizing a human cell without having the right tool that enables us to do that. Similarly, it would be incorrect for us to talk about viruses if we're using a light microscope, because a light microscope is not capable of seeing a virus. Viruses are too small. So in lab, you've discussed microscopes by now, and hopefully you can appreciate the relative size of some of the structures that we're starting to learn about. Now, cells have a challenge, and this is true of every single cell there is, and it's always been true. It's not new. It is a challenge that has been there since all of evolution. So cells must be big enough to hold everything they need. Right? There's a lot of stuff that has to get crammed into that cell. But they also need to be small enough so that they can interact with the environment across their membrane. So they have to be big enough to hold the DNA, the various proteins, and whatever other structures that particular cell needs in order to live and to reproduce, all while being small enough to allow for good interaction with the environment. So that brings in a concept called or referred to as the surface area to volume ratio. And what this is, I'll show you some examples, but what this is it's an evolutionary adaptation that has occurred over many generations, where cells have adapted. They figured out a way to deal with this size problem. And it's actually quite simple. So we see this core theme of maximizing the surface area to volume ratio throughout all of biology. So let's apply this to a simple example, a non-biological one, if you will. So we've got a box here, and this particular box is a 3 units by 3 units by 3 square. So that would be a total volume of 27 units cubed, 3 times 3 times 3. Our total surface area would work out to be 54 units squared. And if we divide the surface to volume ratio, it comes out to 2. But let's look over at the right side of the slide. So now we're looking at individual boxes occupying the same total volume though, right? So we've got 1, 2, 3 boxes by 1, 2, 3, and then they're 3 deep as well. So our total volume is constant. It's still the same as it was because the volume is not changed, 27 units cubed. However, the surface area is much bigger now. And if we look at the ratio between surface area to volume, we have 6 as opposed to 2. So let me give you an example of how to think about this. So imagine it's time to wrap presents for somebody, and you've bought them some type of gift that has 27 different pieces in it, right? 27 components, all the same size. So you could wrap them individually, all 27 pieces individually. It might look like the person's getting more of a present that way because they get 27 things to open. Or we could stack them all in a single box nicely and wrap that one box. So let's think about the surface area of wrapping paper that would be needed in both of those examples. So if it's a single box, we only need 54 units squared of wrapping paper because we only have 6 sides, right? 4 around and then the top and the bottom. However, in the example on the right, we need quite a bit more wrapping paper because we still do have the same 2 sides to every cube, but now we have all these internal surfaces. So imagine how much more wrapping paper would be needed to wrap all 27 boxes separately and then stack them together. You've got all those interior surface areas that would not be present in the single box. So I think that that's a pretty good example of surface area to volume ratio using a real life example outside of biology. So cells have evolved to deal with this by a very simple process. They literally have wiggles in their membrane. And that increases how much surface area there is, right? If I have a spring and pull it out, you can imagine a spring, if I stretch it out, I've got some room to stretch it out. But if it's all condensed down, that's different. So again, cells have this fundamental problem of needing to be big enough to hold everything in them, while also being small enough to allow exchange through the membrane. So we call that either the cellular membrane or the plasma membrane. Those are the same things. It refers to the outer membrane of the cell. So our plasma membrane is flexible. It's not rigid at all. Plant cells have some rigidity in them because they have a wall around them, but our cells don't have that, and that allows them to be more flexible. Now, the membrane is made up of out of phospholipids, which is one of our types of fats. It's one of the macromolecules, and it's always two layers. It's a bilayer. And you can see that we've got some different sections of it. So the gray heads are water soluble, and they are on either side of that membrane. And that makes sense because the inside of our cells are majority water, but so is our body. So the outside of the cells is a watery environment too. So the two surfaces that come into contact with that polar watery environment of our body and cells match it, right? The hydrophobic tails are sandwiched in between those hydrophilic heads, and that essentially protects them. Let me explain this in a different way. If the membrane were all water soluble, if it were all hydrophilic without the fatty acid tails, there would be no membrane. It would just dissolve in the environment of our watery body. If it were all just tails, the phospholipid tails, it wouldn't interact properly with the rest of our body since it's so watery. Remember, like dissolves like. So the combination of having the hydrophilic heads on either side with the hydrophobic tail sandwiched in between them creates the perfect structure for our membrane. So it is described as a phospholipid bilayer because there are actually two layers of it. So those heads are outward and the tails are inside, where they are protected from the watery environment. Now, also in our cell membrane or plasma membrane are a number of different proteins. What we looked at just a moment ago was a very simplified structure of the membrane. But there are many proteins embedded within the cellular membrane, and proteins have different jobs. So we have a number of channels, and I like to think about channels like a tunnel, so one thing can pass through. We also have what are called pumps, and we'll talk more about pumps when we get to energy. Pumps require energy, okay? So that's why we're going to talk about them when we get to that chapter. And then we've got some other structures, other proteins that are present in the cellular membrane as well. And hopefully you're starting to appreciate now that if we have a different protein, it has a different structure. That means it has a different function too. So all of these different proteins that are embedded in the phospholipid bilayer, they're necessary because they have different functions. So this is perhaps a more accurate way to look at it. So we've still got our phospholipid bilayer that we saw before. We've got our internal environment inside the cell versus the outside environment, which would be just the fluid that our cells are bathed in. But now you can appreciate that there are some proteins embedded within this layer. So this channel protein, notice that it spans the entire membrane, and there is a tunnel in the middle of it that allows for fluid to move and whatever is dissolved in that fluid. So our channels are actually specific. These are proteins. So we have a channel for water. We have a different channel for other substances. Looking over towards the right side of the figure, we have other examples of proteins. And some of these don't have regions that interact with the phospholipid tails, the tail section of the bilayer, whereas others go all the way through. So what is typically true of the proteins that are embedded in our plasma membrane is that they have regions that are fat soluble and they have regions that are water soluble. Well, the water soluble regions are going to be in the watery environments, whereas the fat soluble regions are going to be more interior to the phospholipid tails. And every time we have a different protein, we need to realize that there's most likely a different function associated with that protein as well. Or else why would we have different proteins? Now, our prokaryotic cells are much simpler. Every single cell, though, does have a plasma membrane, prokaryotic or eukaryotic. They have DNA, they have ribosomes, which are needed to make protein, and then they have that fluid component, the cytosol. We've got our prokaryotic cells, which are simpler. They're smaller on average, and then the eukaryotic cells. So our cells are eukaryotic. The cells of a bacteria or archaea, they are prokaryotes. Okay, got ahead of myself there. So our eukaryotic cells are distinguished prominently by the presence of a nucleus. And that means there's a membrane around it. So all of the eukaryotic organelles do have membrane around them. And we call this around the nucleus, we call it the nuclear membrane. Sometimes it's called the nuclear envelope. But it does define the nucleus. And depending upon which type of eukaryotic cell we're talking about, there are a number of other organelles that may be present, and they have different functions. So our prokaryotic cells are typically about 1/10 of the size of a eukaryotic cell, and they are much simpler too. So we're looking at kind of a cartoon drawing here of the eukaryotic cell on the left versus the prokaryote. I do want to point out that the relative size between the eukaryote and the prokaryote is not accurate here. The prokaryotic cell should be quite a bit smaller than the eukaryote. But look how much more complexity is present in the eukaryotic cell. Without even knowing what these different structures are yet, you can see that there are more things present in the eukaryote. Over here in the prokaryote, we have those basic things. We have ribosomes. There is a cell membrane, which is shown in black here. But beyond that is a cell wall. And cell walls have more rigidity to them. They're stronger. They provide structure. And then we've got, even beyond it, there is a capsule present around this prokaryotic cell. And we'll talk about these other features. So the inside of both the prokaryote and the eukaryote is correctly called the cytoplasm. And then within the cytoplasm are the different organelles. Now, in a eukaryotic cell, we do not include the nuclear material in the cytoplasm. So it just goes from the wall, or excuse me, from the cellular membrane to the nuclear membrane. I'll point that out in a picture. Whereas in prokaryotic cells, there is no nuclear membrane, there's no nucleus, so it is one bag. We call the region in the prokaryotic cell where the DNA is concentrated, it is termed the nucleoid region, which means it's like a nucleus, but it's not. And the reason why it's not is because there is no membrane around it. In order to be called a nucleus, there must be a nuclear membrane. So in the prokaryotic cell, there is no surrounding membrane around the DNA. It's just in there, all in one bag. Our prokaryotes have that cell wall, and it is much more rigid than the cell membrane. And depending upon what type of bacteria we're talking about, there may be some differences in the components of those cell walls. But all cell walls function to provide protection. They also give shape and structure to the cell. Some prokaryotes have structures outside of their surface. So we see structures like fimbriae or pili, or even flagella. You're probably familiar with a flagella. We have one type of cell that very characteristically has a flagella on it, and that would be the sperm cell. So here's a flagella. So this particular prokaryotic cell has only one flagella, but other types of bacteria may have 2, there may be 50, there may be 100 of them. So please don't think that there's only one flagella. It depends upon the specific type of cell we're talking about. But look at some of the shorter structures, the fimbriae. So the flagella is actually for movement. It's connected to an energy source so it can rotate like a whip and help the cell swim. That's why the human sperm cells have a flagella on them, because they need to move through the liquidy environment, getting to where the female egg is. So bacteria that live in watery environments, they have flagella. Those that say live on our skin, there's no need for a flagella. They don't have them. But the fimbriae, I like to think of them like sticky structures. They help the bacteria or the prokaryotic cell stick to whatever it needs to support its life cycle. So here's the nucleoid region, which is where the DNA is. There's no membrane around it. We've got our ribosomes that are present within the cytoplasm. And then we've got our membrane, our wall, and then the capsule on this prokaryotic cell. There are types of prokaryotic cells that do not have capsules on them. So again, I don't want you to perhaps misinterpret that every prokaryotic cell has a capsule. That's not true. It depends upon which type we're talking about. All right, so now we're going to move to eukaryotes. And for the rest of this module, that is what we will be discussing. So the eukaryotic cell is distinguished by the presence of that nucleus with its nuclear membrane. And then depending upon which type of eukaryotic cell we're talking about, there are a variety of other organelles. And the Latin translation for organelle, L means little organs. That makes sense. So these are the organs of the cell. Every time we see the organelle, the mitochondria, it has the same job. Every time we talk about the endoplasmic reticulum, it has the same job, regardless of the type of cell we're in. So the different organelles of a eukaryotic cell, and there can be many, are divided nicely based upon function. So the way that your textbook and the way that I'm going to teach them to you is divided into, these organelles are divided into 4 groups based upon function. So our first group are the organelles that are involved in genetic control of the eukaryotic cell. This would be the nucleus and the ribosomes. Now, I should emphasize, this is a good time, ribosomes don't have a membrane around them. So technically they're not considered an organelle, but they're often described that way because they're present in the cell, but they don't have a membrane around them. Our second category of eukaryotic organelles are those that are involved in the manufacturing, the distribution, and breakdown of different molecules that the cell needs. And there's quite a few organelles that fall into this category. This is the endomembrane system, which is composed of the endoplasmic reticulum, the Golgi, lysosomes, vacuoles, and then peroxisomes. We've got our organelles that are associated with energy conversions. There are 2. We've got our mitochondria, which are present in all cells. and then the chloroplast, which is present in plant cells. So even plant cells have mitochondria. That's a common misconception, but they have chloroplasts and they have mitochondria. And then we've got the organelles that are involved in providing support to the cell and helping it to move and communicate with other cells through what's ever attached to that cell. So that would be the cytoskeleton. It does include the plasma or cellular membrane. And for cells that have a wall, such as a plant, it would include the cell wall. So here's a cartoon of a eukaryotic cell. And first off, there's a lot more stuff in here. So you can see it's all labeled. I've given you a couple different figures to look at. We'll go through each of these structures as we progress through the content of this lecture. All right. So most of these cells that are plants and animals share most of the same organelles, but not all. There are a few that are present only in plants, and there are a few that are present only in animals. So both lysosomes and centrosomes, these are found in animal cells. You don't find them in plant cells. And that means there's no function for them in the plant cell. And then within the plant kingdom, only a few cells now in our modern plants have sperm on them. Yes, our ancient plants back in the early days of the earth, they were all in water. They needed flagella, they needed sperm for reproduction. But now that plants have moved out onto land, it's different. This is not a watery environment. You know, land is a dry environment. So instead of using a flagella on a sperm, the plant uses something like a pollen grain or a seed. Okay, so now in plants, we've got three main structures that are found in plants, but not in animals. The cell wall is one of those. And the plant cell wall is made up of the polysaccharide cellulose, which is a very strong structure. Within the cell wall are holes. They're called plasmodesmata. And these are channels that allow material to move from one cell to the other. We'll look at a picture. Plants have chloroplasts, which facilitates photosynthesis. And then they also have what is called a central vacuole. It's like a big balloon. It's like a big bladder, but it holds water in a plant and whatever other chemicals are dissolved within that water. So here is another generalized structure of an animal cell with everything labeled. And notice that it's essentially oval shape. It doesn't have a nice brick-like structure. And these are the figures, these are the figures that come out of your textbook. So every time you see the nuclear envelope, it's shown in purple. The endoplasmic reticulum is in blue, and so on and so on. So it's nice to recognize that color coding. But this is the plant cell. So it's more like a brick, and it's because of the plant cell wall. So these little channels, these pores or holes, those are the plasmodesmata that allow for the movement of material from one plant cell to another. But many of the internal organelles are the same. We do have a mitochondria. Plant cells have mitochondria too. But importantly, they have the chloroplast, which is the organelle for photosynthesis. We'll talk about that a little bit throughout in a subsequent chapter. So looking again at a eukaryotic cell versus prokaryotic cell, hopefully you're now able to appreciate the complexity of the eukaryote, but also you'll recognize the basic features that are shared between every cell. And that means that it's able to live. We talked about this in module 1. In order for something to be living, it has to be able to reproduce. There has to be a flow of information. There's interaction with the environment. So we've got those different criteria that define life. Okay, now we're going to move into that first functional group of organelles for the eukaryotic cell, those that are involved in genetic control, namely the nucleus and then the ribosomes. So in the nucleus is most all of the DNA. That might sound weird to you. I said most of the DNA is in the nucleus. Well, you might be thinking, well, where else would there be DNA present in a eukaryotic cell? There's actually two other places where DNA can be found. And we'll talk about that at the end of this module. But most of the DNA that is there. And we know that DNA is the control. It's the powerhouse that directs all protein synthesis. And it does it through that intermediate molecule called the messenger RNA. I like to think of messenger RNA like a translator. I mean, if I speak the English language, I speak French, I don't know how to speak Spanish. So if I'm trying to communicate to somebody who only speaks Spanish, I need to use a translator. Well, that's the job that messenger RNA does in the production of proteins coming from the DNA sequence. So DNA does have a number of proteins that associate with it. And it helps the DNA to wind down in an organized function. If we actually took our DNA molecule and stretched it out, it would never fit inside the cell. We couldn't just like smush it in there. It has to be compacted and folded down in a very specific way. And it does that by creating what are called chromosomes. So our DNA uses a number of proteins to help organize it, to help condense it down to the tight form of the chromosome. Now, chromatin, a little bit different from chromosome. Chromatin refers to the presence of the DNA with those proteins, right? So those folding proteins have to be in there as well. But the only time we see DNA organized as chromatin is when the cell is not dividing. In order for the cell to divide and replicate, the DNA has to unravel so that it can actually be seen and copied. So when it's packed down in the form of a chromosome, which is often how we talk about DNA, that cell's not replicating because the DNA is not accessible to be copied. So you can look at the amount of chromatin present in a cell as a way to determine how active that cell is. All right, so the nuclear membrane, just like the plasma membrane, is a phospholipid bilayer. So it's a double layer, just like the cellular membrane. It does have to have pores in it though, and that allows for the movement of certain material in and out of the nucleus. So the nuclear membrane does regulate what moves in and out of it, and it does this by connecting to what comes next, the endoplasmic reticulum. Now, the nucleolus is a structure only of eukaryotic cells because it's present in the nucleus, and prokaryotic cells don't have nuclei, so therefore they don't have a nucleolus. And what the nucleolus is it's the site where ribosomal RNA is being synthesized. In other words, it is the site where the DNA, let me back up. It is the site where the ribosomes are being made. Ribosomal RNA is involved in the production of ribosomes. So this is the nucleolus, this feature here. So the left side of it is a cartoon that's been drawn. This is actually an electron micrograph that has been stained. So it's a true picture of of this nucleus. So we've got our nucleolus, which is where that ribosomal RNA is being made, and it is surrounded, we've got our DNA around it, and then you can see the nuclear membrane or nuclear envelope. And in it are pores that allow for the movement of material out of the nucleus into the endoplasmic reticulum, or even into the cytoplasm. So the nuclear membrane connects directly with the endoplasmic reticulum. It is a direct connection, and you can even appreciate the color changes as we move through. Now, ribosomes are made of their own special RNA. It's called ribosomal RNA. And depending upon if we're talking about a eukaryote or a prokaryote or archaea, the 3 domains of life, there are distinctly different ribosomes used by the members of the individual domains. So our ribosomes are very different from the ribosome of a bacteria. And the job of any ribosome, regardless if we're talking about a prokaryote or a eukaryote, is to make protein. That's the job of all ribosomes. And it does this through that intermediate of the messenger RNA. So cells that are making many proteins, they have more ribosomes than do cells that only make a little bit of protein, because the ribosome is the site of protein synthesis. Now, they're found in 2 places, ribosomes. They are free in the cytosol, as we saw, but there are also ribosomes that are bound on to the endoplasmic reticulum or the nuclear envelope. Let's look at those. So we've got our free ribosomes here, just suspended, but then on the endoplasmic reticulum, these little brown structures here, you can see it in the actual electron micrograph really well, the brown structures. These are ribosomes that are attached on to the endoplasmic reticulum. So these are bound ribosomes. So we have free ones and bound ones. They do the same job. They make proteins. All right, so we've reviewed the 2 structures associated with the genetic control of the eukaryotic organism, which are the nucleus and the ribosome. We're now moving on to our second category, which are the organelles that are involved in making different materials and processing them and moving them around to the cell, different areas of the cell. So that is the endomembrane system. And it's got quite a few members. So one of the features about the endomembrane system is that some of the membrane is actually connected, like we saw with the nucleus to the endoplasmic reticulum. There's a direct connection in that membrane. So they're physically connected. Other members of this endomembrane system, there's not a direct connection in the membrane. Instead, they use what are called vesicles. And this is a little temporary sac of membrane that holds something in it, and it's moving somewhere. It's like a transfer bubble. So the endomembrane system, the main functions of it as a system is to synthesize different materials that the cell needs, to distribute them, to store them, even to export some of these materials outside of the cell. And also another function of the endomembrane system is it helps to get rid of some materials that the cell doesn't need anymore. So it includes, we've got many members. We've got the nuclear envelope, we've got the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles. The cellular or plasma membrane itself is also part of this system. Oops, sorry, I went too fast. Let me go back a minute. So we've got 6 members of the endomembrane system. And some of these are directly connected membrane to membrane, whereas others are not. I do want to emphasize that not every eukaryotic cell has all of these components, because vacuoles, for example, are found in plant cells, but not in animal cells. And lysosomes are found in animal cells, but not in plant cells. So the ER is the biggest component of the endomembrane system. And we actually have two types of it. So we have what is called the rough ER, and then there's the smooth ER. The difference is the smooth ER does not have ribosomes on it, whereas the rough ER has ribosomes bound to it. That means it's functioning to make proteins. The rough ER is involved in helping to make proteins. That is not the job of the smooth ER. There's no ribosomes there, so therefore it cannot make protein. It has different function. So the smooth ER is very important in making lipids. It's also important in detoxifying our body from various toxins. Where the rough ER makes more membrane, and then it has those ribosomes on it, which means that it's involved in making proteins. And specifically, it makes what are called secretory proteins. So these are proteins that the cell releases outside of the cell. They're not proteins that stay inside the cell. They are secreted proteins, which means they leave the cell. So looking at our rough and our smooth ER, so we've got our rough ER that is studded with ribosomes, but we've got smooth ER, shown out here, that does not have any ribosomes on it. And the rough ER and the smooth ER are directly connected, membrane to membrane. Okay, so let's take a look at what this function might be, an example of this function. So we're looking at the rough endoplasmic reticulum here, because here it is, here's the ER, and we've got a bound ribosome. So that ribosome is going to make a protein, which then is pinched off in a transport vesicle. So what happens is some of the membrane from this rough ER is actually lost. It pinches off surrounding this protein, which now is going to go to the go out to the cellular membrane where it can be secreted outside of the cell. So the ER loses some membrane every time a vesicle is formed, because the vesicle is surrounded with that pinched off membrane. And if you remember when we were learning about proteins, they have their different layers of folding in order to become functional. So we're starting to see some of the folding and also other changes to the protein. In this case, it's called a glycoprotein because it's got a sugar group, glyco for sugar, added onto it. Okay, so we've talked about the nuclear membrane, the rough ER, and the smooth ER. Next comes the Golgi apparatus. And I think of the Golgi like a giant post office. And I don't mean just your little neighborhood post office that might be in your neighborhood. Think of it like the big post office. I live in Rancho Bernardo, San Diego, which is where the big post office is for San Diego. So all the mail that comes in with a zip code for San Diego goes there. Or maybe there's another big one that I don't know about, but a lot of it goes to the Rancho Bernardo facility. So it goes in there and then it's sorted. It's sorted by zip code. And then the mail with one zip code is sent to a local neighborhood post office, or it's sent out on the mail trucks and delivered to our house. So that's a good analogy for the Golgi apparatus. It has a receiving side where material comes in, but then it also has a distribution side that sends out the materials the cell needs. So that's the job of the Golgi, is it helps to send materials, pack them up and send them to their right places. It puts tags on materials so that the cell knows what to do with it. So we've got the receiving side. So again, the post office that receives all the mail for San Diego goes in the receiving side, and then it's processed, and then we have our shipping side. Let's look at that. So here's the Golgi apparatus. It's shown in your textbook in this kind of pinkish orangey color. So we begin with a transport vesicle that has been released from the endoplasmic reticulum. So it contains some type of secretory protein in it. So we're picking up where we left off before. You can see it's blue because the membrane comes from the ER. It binds, it fuses with the Golgi membrane. So you can see that little bit of blue here. So the Golgi receiving side actually gets bigger in membrane size because it has incorporated that membrane of the vesicle. And in doing so, it spills out the secretory proteins that are here so that they can be properly modified and tagged and then sent across the Golgi to the shipping side where they then pinch off in another vesicle. But notice now the membrane comes from the Golgi. It's the same color as the Golgi. Next, this transport vesicle is going to go to the cellular membrane where it can release the materials. So lysosomes are digestive compartments. It's containing enzymes, acidic material that help to break down materials. So a lysosome is like a chemical digestion. It is made by the rough endoplasmic reticulum, so that's part of the job of the rough ER, but they are processed in the Golgi, right? Because that's the job of the Golgi, is to really process everything that's moving around in the cell. So what are the functions? Well, sometimes they are used to digest food. We'll look at an example, but they can also be used to help with our immune response and with getting rid of damaged old cells. They do this by spilling their enzymes out. We have other lysosomes that function to help get rid of different materials that have become damaged in the cell. And that way the cell doesn't use them anymore and can replace them with normally functioning materials. So this is an example of a food lysosome. So we've got the lysosome containing its digestive enzymes made by the ER, and our plasma membrane is shown here in gray, and some food has come into a vesicle that has pinched off from the plasma membrane of this cell. So now it is properly called a food vacuole, and it's bigger. Vacuoles are bigger than vesicles. What happens is the lysosome is going to fuse, so the two membranes actually become one, and that spills the digestive enzymes of the lysosome onto the food, where the food is digested down to its pieces that the cell can then use for whatever it needs. So this is an example of a lysosome fusing with a food vacuole to digest food. The example of removing a damaged organelle is shown here. So we've got a vesicle that has a damaged mitochondria in it. The lysosome fuses, spilling the digestive enzymes onto that damaged mitochondria, which is then broken down into small enough pieces that the cell can deal with them. Go ahead and play this video for you on how lysosomes form. But keep in mind, try to think a little bit more than just lysosomes and orient yourself with the other members of the endomembrane system that you have learned about now.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and moved through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
So you can see we've got movement of the material out of the shipping side of the Golgi apparatus, which will allow the digested material. Well, we're forming the lysosome, which then will go on and fuse with whatever it's digesting. Okay, so talking about vacuoles, the vacuoles are all about water balance. And these are structures that are not in animals. We don't have them. They're found in plant cells, but they're also found in protists, which are an interesting group of organisms. So protists may have what are called contractile vacuoles, and they help the organism to get rid of water. They help to like spit the water out of the organism. But in a plant, the vacuoles can have different functions. They're involved in digestion of the plant food. Some of them have pigments. Some of them even have the toxins that a plant produces. And this way, the plant is not susceptible to its own toxin because it's contained within the safety of a vacuole. So something like poison ivy, for example. So we're looking at a protist here. This is called Paramecium, and we've got two examples of contractile vacuoles that are present. And again, these work by helping to eliminate water, which propels this organism. It helps it to, actually helps it with its movement. Okay, so now we're gonna put together all the members of the endomembrane system. It begins in the nucleus, which means there is a nuclear envelope. That is what defines the presence of a nucleus, having that membrane around it. The nucleus has pores, which allow material to move into the cytoplasm or into the endoplasmic reticulum. So we go from the purple color of the nuclear membrane to the blue of the endoplasmic reticulum. The part of the ER where there are ribosomes bound to it is the rough ER, but we've also got the smooth ER that does not have ribosomes. So we've got a protein here that has been produced, a secretory protein that has been produced with the association of the ribosomes of the rough ER, and it has budded off in a transport vesicle. So the vesicle has the same color membrane as the ER because that's where the membrane came from. It comes over to the Golgi apparatus and fuses. So you can see the blue fusing in with the color of the Golgi, and that spills the contents of whatever is in that vesicle into the Golgi where it is processed. If there need to be modifications made to it, that can happen, such as a sugar group being added to it. It can have different tags added to it. And when that's all said and done, the material pinches off in what is now called a second transport vesicle. But notice the membrane's a different color than our first transport vesicle. And finally, the transport vesicle will go to the cell membrane and fuse to it, allowing the material to be spilled into the extracellular environment. So the cellular membrane gets larger each time a transport vesicle fuses with it, incorporating that membrane. So the various organelles of the eukaryotic endomembrane system are either directly connected through membrane to membrane or indirectly through the formation of vesicles. But this system works to manufacture, process, and move the different materials around that our cells need. Now, peroxisomes are also part of the endomembrane system, but they are, but they aren't. They're kind of in their own unique category. They're not part of the endomembrane system because they don't come from any of the organelles that we just talked about. So they're kind of unique in that we we don't know much about them. But they are important in helping to break down fatty acids. And that means that they are used in the generation of energy or fuel for our cells, because fatty acids are a good fuel. All right, we're going to move now to the third category, functional category of eukaryotic organelles. And those are the two organelles that are involved in energy: the mitochondria, which every cell has, and the chloroplast, which some cells have. Plant cells have them. So the mitochondria is all about energy. It is the powerhouse. It is the location of cellular respiration. It is where cellular respiration occurs. And that essentially is metabolism at the level of the cell. We're going to study that in our next coming chapters. So what cellular respiration does is it takes the energy from our food, so it's in the form of a chemical, and it converts it to this other molecule called adenosine triphosphate, which is abbreviated as ATP. It's energy. So we spend the currency of the of the United States is the dollar. The currency of the cell is ATP. Now, our mitochondria have different compartments, and this is so important for structure and function. It's a great example of that relationship between structure and function. So it has an outer membrane and an inner membrane, and then in between it is an inner membrane space. And this functionally is a really important location because it is where the proton gradient is generated. We will learn about that when we move into our chapter on cellular respiration. But since we're learning about the different structures, I wanted to just mention that the function of the structure. So we've got our two membranes with the inner membrane space in it. And then within that inner membrane, we have the matrix. And there is actually DNA present. Mitochondria have their own DNA. They have their own ribosomes, and they have enzymes that are necessary for energy conversions, for cellular respiration. So at the beginning of this module, I mentioned that most of the eukaryotic DNA is found in the nucleus. Most of it is, but some of it's here. Mitochondrial DNA is only present in the mitochondria, but it is DNA. It has its own ribosomes. That's interesting. Why would the mitochondria need to have ribosomes, since ribosomes are associated with making proteins? We're going to come back to that. It has many enzymes. Wow, it really sounds like the mitochondria is a pretty self-sufficient organism. It's actually much like a prokaryotic cell. It has what's needed to be a prokaryotic cell: DNA, ribosomes, and a membrane, plus the fluid. Okay, so the inner mitochondria membrane has folds in it, and those are called cristae. And this is that simple evolutionary adaptation that we began our discussion with, where we have wiggles or folds or more boxes to wrap that increase that surface area while holding the cell size constant. This is a way that a cell is able to still be small and contain everything it needs and function well with the environment. Because that membrane isn't just a nice smooth circle, it's a wiggly, 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 wiggly membrane, which means there's more of it, which means that whatever cellular machinery is present in that membrane, if we have more of that membrane, we have more of that cellular machinery, which means the mitochondria can do its job better because it has more equipment to do it. All right, so this is the mitochondria, and they always look like this in your textbook. Same color, same shape. So we've got our outer membrane, which is the smooth one, and then here's that inner membrane. So the folds are called the cristae, but they are only present they are a part of the inner membrane. The membranous space, that inner membrane space, is between the outer to the inner, and we're going to learn how important it is functionally. And I guess that's really all I wanted to point out about it, but look how extensively folded that inner membrane is. So if we took the outer membrane and stretched it out, just say it's this long. We take that inner membrane and stretch it out, it's much longer. So that means that we've got more material to do that job. That's what the folds do. This is an evolutionary adaptation to maximize the surface area to volume ratio of the cell. And we see this over and over again in evolution. And that tells us it works. I mean, when we see the same structure serving the same purpose, it tells us that it's a successful structure and successful function. So, all right, so photosynthesis is associated with plants. Now, some bacteria and algae are capable of photosynthesizing too. So that means they have the same equipment somewhere. Bacteria don't have organelles. They don't have a chloroplast, but they do have that machinery present in their cell membrane for bacteria that are capable of doing photosynthesis. So the chloroplast is the site of photosynthesis for plants and algae. So algae have a chloroplast, bacteria don't. Algae are eukaryotes, bacteria are not. Just like we saw with the mitochondria, the chloroplast has some pretty similar features. It has different compartments, which means different locations, different jobs, and those different locations can specialize their job. It too has an inner and outer membrane with the inner membrane space. But in a plant, in the chloroplast, we call that fluid the stroma, and it's thick like jelly. The wiggles in the chloroplast are thylakoids. In the mitochondria, they are called cristae. In the chloroplast, they are called thylakoids. Those are analogous structures. And I think of a thylakoid, I'll show you, but they look like a pancake. And then a stack of them, a stack of pancakes is called the granum. Let's take a look. All right, so one of these is a thylakoid, and then one pancake is the thylakoid, but the stack is the granum. We've got our outer membrane and our inner membrane, the inner membrane space, but then the thylakoids are the equivalent internal wiggles of that inner membrane. So a division of location and work. And each plant cell has many chloroplasts. Each cell has many mitochondria. These figures are simplified just to make it easy to look at, but I don't want you to think that there's only 1 or 2 chloroplasts or mitochondria per cell. There's many. So which membrane in the chloroplast appears to be the most extensive, which has the largest surface area? And this would be the thylakoid, which is analogous to the cristae of a mitochondria. Why? Well, this is the location where photosynthesis occurs. The machinery that is needed for photosynthesis is located here in the chloroplast. So if we have more of the thylakoid, that means there is more opportunity for photosynthesis to occur. All right, so mitochondria and chloroplasts are very interesting. Like the mitochondria, chloroplasts have their own DNA too. They have their own ribosomes. So this brings us to what is called the endosymbiotic theory. And it's a theory that explains how the evolution of the prokaryotic cell as it got more complex, but also how the first eukaryotic cell came to be. And it's actually quite simple. One bacteria ate another. I'll show you. So mitochondria and chloroplasts, they have their own DNA, they have their own ribosomes, and they have their own enzymes. The structure of the ribosomes that are present in the mitochondria and the chloroplast is the structure of the prokaryotic ribosome. It is not the same structure as all of our other ribosomes, which are eukaryotic. So this tells us that the mitochondria and the chloroplast most likely were bacteria at some point prior to the evolution of eukaryotes. So the endosymbiotic theory states that the mitochondria and chloroplasts were originally a prokaryote, an independent prokaryotic organism that was consumed by another cell, creating a eukaryote. So think of Pac-Man eating something, eating another cell. So here we go. So we've got our nucleus, we've got our endoplasmic reticulum. So we're actually looking here at the primitive eukaryotic cell. Right, it's correct to have a nucleus and ER. So what happens is this primitive, this ancestor ate a mitochondria, which was originally a bacteria or archaea or prokaryote. And it evolved to become the mitochondria over many, many, many generations of time. And then we had a second event where another prokaryotic organism was engulfed. But this time it is one that was doing photosynthesis. And this is the theory of where the chloroplast came from, is engulfing a photosynthetic prokaryotic organism. And that brought those structures and consequently the functions into the photosynthetic eukaryotic cell. So we have two different events of a prokaryote being engulfed by this eukaryotic cell, creating the non-photosynthetic eukaryotic cell, such as ours, as well as the photosynthetic eukaryotic cell that is found in plants and algae. All right, this brings us, so we've now wrapped our third category based on function, which are the organelles that are associated with energy conversions. Those are the mitochondria and the chloroplast. That brings us to our fourth category of eukaryotic organelles, the cytoskeleton and the different projections on the outside of the cell. So the cytoskeleton, just let's look at that word. Cyto means cell. Skeleton, well, you know, we have a skeleton, you know what that is. So the cytoskeleton, these are just different proteins that provide structure to the cell. We have three main types of cytoskeleton material, three fibers. The first is called a microtubule, and it's made of a type of protein called tubulin. Tubule, tubulin. They're small. So they do provide shape and support to the cell. You can think of them like a train track. They help to guide material through the cell. And we will see how this, we'll really dig into this more when we start studying cellular replication. So in our cells, they come out of an area called the centrosome. The second type of fiber is the intermediate filament, and it's in most every animal cell. And it really helps to hold some of the organelles in place. So it's providing some shape as well. The intermediate filaments don't move around. They're not temporary structures. These are usually present all the time. And then our third fiber are the microfilaments, and these are made out of a different type of protein called actin. And they give shape. They also help with movement. All right, so now we've got all three types lined up. We've got our microtubule, which is made out of tubulin, and look at the structure. So it's like, it's got an internal hole in it, and it's wrapped. Compare that to the intermediate filament, which is much smaller, by the way. The intermediate filament is quite a bit stronger because it's not just one chain wrapped around a hollow center. The microfilament is like a rope or a guy wire that has multiple chains wrapped around each other, and then they're wrapped around each other and around each other like rope. So it makes it quite strong. And then our microfilament really has kind of an intermediate. It's got features of both of the other two filaments, but it is the smallest, and it's made out of one type of protein called actin. So now let's look at the pictures, the micrographs above. On the left, where we're looking at the microtubules, all of the structures in green are microtubules. So you can see they're present all over the place. The blue is the nucleus. So pretty much microtubule is present everywhere outside of the nucleus. And then looking at examples of the intermediate filaments, in this picture, the nucleus is orange, but you can see the intermediate filaments around it. And then our microfilament is the actin protein, which is shown in red here. So different proteins have different structures, and they have different functions. But overall, the cytoskeleton provides structural support and integrity to the cell, while also allowing for the anchoring of materials and movement of others. So this is actually a little preview into where we're going in our genetics section. But I put this in here because it's showing you the microtubules, showing you some of these filaments that are produced. So this is the centrosome, which is where these tracks originate from. These are actually duplicated chromosomes that are being pulled apart into what will become two different cells. But see how each chromosome has a track? It's guiding the chromosome so that it goes into two separate cells, first of all, without getting tangled up on everything else that's in the cell. So what we're looking at here is this protein. All right, so cilia are other appendages that may be on the outside of a cell. And these are like, I think of cilia like if you've ever been in a rowboat with multiple people, everybody has to row together in order for the boat to move properly, right? If somebody stops rowing on one side, the boat turns in a circle. Or if somebody's paddling too fast, that could affect the direction too. So cilia work by moving together, but they're not really, they don't help a cell move, they help other materials move. What cilia do is they help a cell stick to something, if it's a eukaryote, and then they help to move other material around. So our respiratory tract is lined with cilia, and that helps if we get any particulate, like breathing in smoke from a fire or some type of allergen, the cilia should beat up, and they do it all together. And what that does is it helps to move any of those particulates out of our lower airway so that it doesn't cause a problem. Also traps them in mucus too, and then the cilia help to get the mucus out. We've talked about flagella. Flagella are actually movement, okay? Cilia and flagella both need energy, so they are attached to an energy source, but they really have different functions. They both move, but one is talking about moving a cell, where the other is talking about moving something around that structure. So our cells, some of our cells and tissues can have cilia as well. Prokaryotes can have them, but so can we. So this is a picture of stained cilia that is in the human respiratory tract. And you can see, oh, excuse me, you can see how they're all pointing one direction. So feasibly, the direction of any particulate that would get in here would be up and out through a cough or a sneeze. So that's what a cough or a sneeze is doing. It's trying to get something out of our body that shouldn't be in our respiratory system. So the organism that causes strep throat, that bacteria, it actually sticks right onto these in our throat, and that's how the infection is established. Let me go ahead and play this short animation that shows you how cilia and flagella move. Again, they are attached to an energy source because energy costs, or movement costs energy. But they have a shared structure. It's called it looks like a wagon wheel. It's called the 9x2 because there's 9 doublets around and then 2 inside. But both cilia and flagella have this shared structure.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
All right. So we'll come back to this as we move through more of our semester, but you could see that when the red bond moved between these two, that the protein, it caused movement and bending of the structure. So cilia and flagella are similar, although they have different functions. They are found in many different prokaryotic organisms, but they may also be found in some eukaryotic cells or structures. All right, so outside of the cell is what is called the extracellular matrix, and it's actually an orchestrated, tangled network, if you will, of different proteins. So the extracellular matrix is actually what allows one cell to bond to another to form a tissue. It provides integrity to the plasma membrane of all the cells that are anchored through the matrix. And it's also critical in the communication of one cell to another. So, whoops, going too fast. So here's a picture of the extracellular matrix. So we've got our phospholipid bilayer. We've got a couple embedded proteins shown here. And then inside these orange ropes, those are the microfilaments of the cytoskeleton. So they're helping to anchor material. But notice they are attached to the membrane as well. So they are helping to provide support to that membrane. But then everything else on the other side of that phospholipid bilayer is the extracellular matrix that's in the external world. I mean, the cytoskeleton is part of it too, because it's actually attached to these proteins that then attach to these green fibers. So there is a direct connection. So the purple is collagen, which you've heard of collagen, and then the green just represent different other proteins that have different sugar groups on them. We've got engagement of plasma membranes with other proteins that connect them to the matrix too. So our cells form tissues by attaching to each other. And we have what are called junctions, which are the attachment points. So we have 3 main types of cell junctions that are found in animal cells. The first, the tight junction, prevents liquid from leaking out. So I think of it like a super reinforced zipper. You know, you zip it up, it's a waterproof zipper. It's got, right, zippers have lots of teeth that engage. It's not just like a button down shirt where there's a button every few inches. A tight junction prevents the seepage of liquid, of water, and whatever's dissolved in it. We have what are called anchoring junctions, and these form sheets of cells that then form tissues. And then the third type is the gap junction, which is like a tunnel. So it allows for the movement of materials from one cell to the other. So tight junctions prevent movement, gap junctions allow it. So let's look at some pictures. So we've got one cell here, here's another cell, and then a third cell in this picture. So we've got 3 cells, and we've got examples of all 3 junctions. So this is the tight junction, which again prevents the leaking of water. So think of it like a nice tight zipper, but notice how it's extensive. It's not just only located at the points where the membranes join. It goes all through it, providing support. The anchoring junction is shown right here, and these are needed to help deal with things like when our cells expand, right? That could put pressure on the junction and rip it apart, but the anchoring junctions help to keep it there. And then finally, this is an example of a gap junction here and here. And these are little channels, little tubes that allow for the movement of fluid and whatever's dissolved in it to go from one cell to another. Now, I do want to point out, you'll learn about this more in physiology, these channels don't just allow anything to go through them. They're selective. So they're made of different proteins. For example, we have one called aquaporin, water pore. It allows for the movement of water between it, but something else doesn't go through it. They're specific. Now, the cell wall of a plant provides some additional challenges because it's rigid. So plants don't need all that internal cytoskeleton as much as an animal cell does, because they have the rigidity of the wall to give them their structure and support. They definitely have cytoskeleton. I don't want you to think that they don't, but not as extensive as what you see in an animal cell. Plant walls are made out of cellulose, which is a polymer of glucose. And then they have those pores, the plasmodesmata, that allow for the movement of materials from one plant cell to the next. So water, food can move through there, and even different chemicals in the form of messages. All right, so here we're looking at one plant cell, a second, a third, and then part of a fourth. And notice that these plant cells all have essentially the same shape and size, and that's because of the cell wall. It helps to make it more consistent. But here's an example of one of the plasmodesmata. So it's a full tunnel through all of the layers. So we've got the cell membrane and the cell wall that need to be bypassed by the plasmodesmata. So we've got this kind of dark brown layer is a sugar, and then we've got our cell wall, primary wall, and then the secondary wall. So there's quite a bit of structure that needs to be bypassed in order for water and nourishment to move cell to cell. Plants also have the equivalent of veins and arteries, just like we do. So they do have a way to move materials through them. But when we're going from cell to cell, it goes through a plasmodesmata. But this is also a way that water from the environment is able to get in. All right, so that wraps us up. We've gone through our 4 groups of eukaryotic organelles based upon their functions. We've got genetic control. We've got the manufacturing, distribution, and breakdown of different cellular materials. We have energy processing, and then the structural support, movement, and cell-to-cell communication. Those are the 4 categories. So I've included the table from your textbook that summarizes each of the components of those 4 groups, along with a little picture and description of what it does. So I'd encourage you to look over these. And then at the end, I include sometimes the same slide you've already seen earlier in the module. I include them in the back because I think they're really important concepts for you to study. So this is one of my ways to communicate with you that this is really important. If you see a slide again at the end of the module, I'm putting it there for a reason. All right, so the structure and function of the cell of the plasma membrane. And then you've got a diagram that you can label the different organelles of an animal cell. And that's it. All right, that wraps us up on Module 7. Hopefully this recording will come back super quick and I'll get it up to you soon. As always, please come to office hours, and you're always welcome to reach out with email. If you're not free to come during my scheduled office hours, just let me know and we'll set up something. Take care. Bye.